All right. Good morning, everyone. Thank you for being on time. So I am Louise, and I'm joined here with my colleague Ben, and I'll be giving the course today, the workshop today, and Ben will be helping out in the background with any questions. Um, so today we're going to learn about the Ensembl genome browser, and I have a few slides here that I just want to start off with. So at the end of the workshop, I hope that you will be able to describe what Ensembl is, how to navigate the online browser, how to access the different types of data that you can find in Ensembl, and most importantly, where to find documentation and where to go for help. Our workshop structure is going to look like this. So we're going to begin each module with a presentation where I will tell you what the tool or the data is about and how it's being produced or processed. This is followed by a demonstration, a live demonstration on my screen, where I will walk you through how you can get the data on Ensembl or how to use the tool. And then at the end of each module, we will have time for some exercises. So this gives you a chance to try and find the data yourself and try out the tool. And remember that this is not a test. So the exercises are there for you to become familiar with Ensembl and explore the different types of data that we have. So all materials are available on the Ensembl training page. This is under training.ensembl.org, and I'm just going to show you how this looks like. So I'm going to move to my browser on my screen. I hope you can see this. If not, please let me know, and I will restart my screen sharing. So I'm on my browser now. Ensembl works best with either Firefox or Chrome. I'm using Firefox at the moment. And if you go to the page training.ensembl.org, Ensembl without the E at the end, you can find our training page. And this is, here you can find anything related to Ensembl training. At the top of the page, there is a link that says training materials. So I'm just going to click on that link. And our training materials are in this very first link, so the one that says Noblekinmat. So if you open this link, this is our dedicated training page. On the right-hand side, we've got a link to my presentation. So these are all the slides that I'm going to go through. You can download these in your own computer. Underneath, we have a living document. So I'm just going to show you how this looks like. Just opened it in a new tab here. And this is just a really useful document for asking questions. And there, towards the end of the document, there are also some links to some useful resources if you want to go beyond our workshop today. So some learning outcomes in the very first page. If you scroll down, we've got our course overview, the link to the training materials. At the end of the workshop, I'm going to give you some time to leave some feedback for us. And most importantly, on page 4, we've got a section for questions. So if you have any questions, I do invite you to either enter them into the Zoom chat box or enter them in here. Or if you feel brave, you can also raise your hand on Zoom and ask them out loud. I think that the living document is most useful for questions, because in maybe 10 days time, you've forgotten what the answer was to your question, you can always come back and use this as a reference. So this document and all of the training materials that we're going to use today will be available to you indefinitely. So that means even in 3 months' time, if you want to go through the exercises again, you can always come back and do them in your own time. So let me close this living document, and I'm just going to copy the link for the training page into the chat box so that you have it handy for later on. I suggest you keep this tab open on your browser because we're going to come back to it quite a lot. So we've got the materials on the right-hand side. I've just gone through this. On the left-hand side, at the very bottom of the page, we've got our different modules that we're going to go through today. So if you open each of the tabs, it will give you a section for demo. So these are just screenshots of what I'm going to go through live on my screen. And also one section for exercises. So these are the exercises that we're going to do towards the end of the module. All right, so I'm back on my presentation now. Again, if you have any questions, please enter them into the chat box or into the living document that I just showed you, or you can raise your hand and ask your question live as well. And this workshop was also recorded, so I'm currently recording it, and I will share a link with you and Ashraf as well once I've processed the recording. Okay, so our agenda for today, this morning, we're going to go through an introduction to Ensembl, so just a bit of background and history, and I will show you how you can explore genomic regions on the browser, so how to visualize specific coordinates on the browser. This is followed by a 2.5 hour break. And then in the afternoon, we're going to continue on and talk about genes and transcripts, so how we annotate these in Ensembl. And we're going to look at one of our tools, one of our most popular tools, which is BioMart. And BioMart allows you to export large amounts of Ensembl data without the need of knowing any programming language. So it's a very useful tool. And towards the end, I'll just wrap this up. We'll summarize everything we've learned, and I'll give you some time to leave some feedback for us, because this is really helpful to improve our courses for future participants. Okay, so just a bit of background and an introduction to Ensembl. So in 1977, Fred Sanger and his team sequenced the very first genome. This was a bacteriophage. And this really kickstarted the era of sequencing and sequencing more complex genomes. The very first draft of the human genome was published in 2004. So this was a project which started in 1990. And since then, many, many more organisms have been sequenced. And today, we can sequence a complete human genome in a matter of days. So we've come a very long way. And with the sequencing of the first draft of the human genome, Ensembl was launched in 1999 with the goal to automatically annotate this draft of the genome and also integrate it with other data and make it publicly available. But why do we need genome browsers like Ensembl? So sequencing has become much cheaper and faster. And because of that, genomic data is quickly piling up. And when sequencing data is produced, you get something like this. So you just get a bunch of A's, T's, C's, and G's. So trying to read a genome without annotation data would be like trying to read a new language without a dictionary. So it wouldn't really mean anything to you. And this is exactly what genome browsers are there for. So genome browsers map annotations and different features to genomic regions, giving the genomic region a value. And what Ensembl does is it brings together information from a wide range of other databases into a single site. Other genome browsers that you might have heard of are NCBI and UCSC. And I also want to emphasize that Ensembl does not do its own sequencing. So we simply take publicly available sequencing data, and we use our own pipelines to annotate the genomic data. We have many different features that we annotate in Ensembl. So these include genes, variants, promoters, binding sites, and many, many more. We have variation data in a number of different species. We've got comparative genomics data, so alignment, gene trees, and homologs. We also have regulatory builds for a number of species. We've got different tools. So one tool we're going to explore today is BioMart, which allows you to export large amounts of Ensembl data. Another popular tool as well is the variant effect predictor, which allows you to find out the consequences of your genetic variants. You can also display your own data in Ensembl, and we provide programmatic access via our APIs. So Ensembl is completely open source. You can use Ensembl data for your research as you wish. And our source code is also available on GitHub if you're interested. There are different ways to access Ensembl data. We are going to focus on how we can access data through the browser. So this is most useful if you're looking at data one by one. But for more advanced users, you can also access data via our APIs and MySQL servers. For those who are not too familiar with programming, we have a tool, BioMart, which you can use to export large amounts of data without any programming knowledge. So this has a user interface which lets you retrieve Ensembl data with just a point and click method. And if you want to retrieve any whole genome data, you can get these from our FTP sites. I will show you how you can access our FTP during the demonstration towards the end of this presentation. Okay, so the Ensembl browser, the Ensembl main browser, is dedicated to vertebrate species. But as we know, vertebrate species are only a fraction of the different organisms in the world. You can find some non-vertebrate species in Ensembl, some non-vertebrate model species, so for example, fly, worm, and yeast genomes, since they're commonly used in research. But in general, if you want to retrieve any non-vertebrate data, you'll need to go to our sister site for this. So this is Ensembl Genomes. Ensembl Genomes is divided up into different taxa. So we've got a portal for plants, a portal for invertebrate metazoa, bacteria, protists, and fungi. And we also have a separate portal for the coronavirus genome. All of these different portals are actually structured the exact same way. So once you are familiar with, let's say, the main site, which we're going to focus on today, you'll have no problem navigating any of these other sites. One thing to note is that that genomic data is constantly evolving and changing. So Ensembl is updated about every 3 months to represent the most recent findings. And behind every update, a lot of work is involved. So this includes adding any new genome assemblies, updating any underlying software, updating gene sets, adding new comparative genomics analyses on genomes and genes, updating variation data, and so on. So the current release we are in is Ensembl 1.11. So this was published just recently in January 2024. And around April, May time, we are expecting to bring out the next release, 112. So because Ensembl is not static, you want to make sure that you write down which Ensembl release you're working with. I'll show you during the demonstration what release you're working with and also how to find archives if you're working with an older release. If you are working with an older release, an older version of Ensembl, we keep the browser interface for Ensembl archives for up to 5 years. So you can still use an older version to keep your research consistent. After 5 years, we retire the browser interface, but you can still access all of the data in our FTP site. So this is just a summary slide of the different Ensembl sites that we've got. So I've mentioned the Ensembl main site. So this is dedicated to vertebrate genomes. The Ensembl genome site is dedicated to non-vertebrate genomes. We also have an Ensembl rapid release site. So there are a lot of sequencing projects around the world going on, and to keep up with the publication of new genomes all the time, we've created the Ensembl Rapid Release, which is updated every 2 weeks instead of every 3 months. And so the rapid release site is has less detailed information on genomes. So compared to the Ensembl main site, so for example, for the Ensembl main site, you would have genome annotation, variation data, and for some species, we also have regulatory data. But for the Ensembl rapid release site, you'd only have annotation data available. So the information on genome annotation is limited here. But if you can't find your species of interest in either Ensembl or Ensembl genomes, you might be able to find it in rapid release. We also have a separate portal for the older GRCH37 human assembly, and we have already mentioned a separate portal for the coronavirus as well. So again, Ensembl rapid release is updated every 2 weeks. The really cool thing about this browser is that we release genomes from loads of different ongoing projects, like the Darwin Tree of Life. So this project aims to sequence all of the different organisms across the UK and Ireland. You also have genomes from the Human Pangenome Reference Consortium. And if you are working with the newer T2T human assembly, you can find this on the Ensembl rapid release site. But just remember that for genomes in the rapid release, only gene builds, so annotation data, and BLAST, and the only other tool you can use is BLAST. Okay, so let's just quickly look at how genome assemblies are made. So the human reference genome is not a genome of a single person, and it's not a consensus genome either. So it's actually made up of fragments from different individuals. And how are genome assemblies generated? So on my slide, you can see a simple diagram of sequencing reads. So each of these lines represents sequencing reads, and sequencing data is assembled by matching up any overlapping reads. So kind of like putting together a puzzle. And what this makes up is the contiguous sequence, or the contig. And you're going to come across this word contig quite a lot in the browser. And this is the main reason why I'm showing you this, just so you know where the word is coming from. So a contig is just a consensus region of DNA. These contigs are then assembled into scaffolds. A scaffold is just a run of contigs or an assembly of contigs without any gaps. And finally, scaffolds are assembled into chromosomes when there is enough mapping information available. So often when you are looking at genome assemblies, you will find that you will find an assembly status, which either says contig, scaffold, or chromosome, and that just describes the quality or how well assembled the sequence or the genome assembly is. So the most complete assembly would have either a full or a chromosome status. And with that in mind, genome assemblies are still not a perfect representation of the genome. So just like earlier versions of the world map, so it's really important to remember that there are still errors and gaps in the genome assemblies. We know that a genome assembly is not a perfect representation of the genome, so there are still gaps, but researchers do fill these gaps and correct errors and share patches and updates continuously. So this means that there can be different versions of genome assemblies. So Ensembl has 4 different versions. So we've got the most up-to-date and supported GRCH38 or HG38 assembly. This is available on the Ensembl main site. We've got the older GRCH37 or HG19 assembly. This is available on a separate portal. So if you're still working with this assembly, you can just go to grch37.ensembl.org. Just keep in mind that this assembly will have greater gap lengths compared to the more recent GRCH38. And there's only limited data and software updates for this. We also have the older NCBI 36 or HG18 human assembly. This is also available on a separate portal, but it has much greater gaps. So there are 150,000 gaps in this assembly, and it is no longer being updated. And the fourth assembly, I mentioned already, is available on Ensembl Rapid Release. So this is the T2T assembly. Oh, sorry. Okay, so what can you do with Ensembl? What can you use Ensembl for? So just to summarize, you can explore genomic regions on the browser. You can find features that you're interested in. You can learn more about genes and transcripts or variants of interest, and also find out more information about the genetic background of a phenotype, for example. So I'm going to move on to our first demonstration for today. So we're going to look at the Ensembl homepage. We're also going to explore the Ensembl sister site, Ensembl Genomes, and learn how to find information about species and genome assembly. Before I switch to my browser, are there any questions? All right. If you do have any questions, please don't feel free to ask them. Okay, so let me move to my browser. And I'm going to open a new tab, and I am going to show you the main Ensembl site first. So simply enter ensembl.org, again, Ensembl without the E. Okay, so this is the Ensembl main site. I'm just going to zoom in a little bit more just so you can see it a bit better. At the very top of the page, we've got the navigation bar. So these just include any shortcuts to some popular links and tools. So for example, we've got links to our BLAST, variant effect predictor, and the other tools, BioMart. Under downloads, you can find out how to retrieve data, whole genome data from our FTP sites. So let's just click this and see how this looks. So I've just clicked on downloads. On the left-hand side, we've got a link that says FTP download. So here in the panel. So if you click this, and scroll down on the right-hand side, you get this huge table. And this table includes any links to our FTP site. So for example, if you wanted to download the entire sequence of the human genome, you can do so by going to this link. So you can download the DNA, cDNA, CDS, and non-coding RNA sequences. You can also download any annotation files, variation files, regulation if available, and so on. So if you click one of these links, so let's just click the human DNA FASTA file, this will bring you to our FTP site. And in this FTP site, you can find the FASTA files for each of the chromosomes in humans. Okay, going back to the homepage, we also have a link to help and documentation. So if you are interested in how the data was produced, or if you're not sure what type of data you're looking at, you can find a lot of help in our help and documentation pages. And if you like, we also have a blog. So in our blog, we always publish, for example, any updates to the newest release, any additions, or even job opportunities at Ensembl. If you use Ensembl quite a lot, I do recommend you register an account with us. So this is completely free. You can register an account and save all of your findings, all of your jobs from the tools in your account to make sure that you can find them more easily later on. Okay, so in Ensembl, you have two different ways to search the website. So you can do a site-wide search using the search bar in the top right-hand corner, or you can use a species-specific search in this blue box here. So if you know which species you're working with, I do suggest that you use this search option because it just makes the search more efficient. Just makes it a bit faster. On the right-hand side, we've got a description of Ensembl in the top. Scrolling down, we can see which release we're currently working with. So this release right now is Ensembl 1.11, published in January 2024. And we've got a few highlights of the newest updates we've done for this release. Scrolling down, we have a link to the Ensembl Rapid Release site. So this is the site that's being updated about every 2 weeks. Let's just see how this looks like. So this is the Ensembl Rapid Release site, the homepage, and you can see that the structure of the homepage is quite similar. It's very similar to the Ensembl main site. So you'll have no problem navigating any of the other portals once you're familiar with one of them. Okay, I'm back on the Ensembl main site now. Scrolling down, we've got some more links here. So these are just examples of how you can retrieve different types of data. And at the very bottom right-hand corner, there is a link that says View an Archive Site. So if you're working with older releases, you can click this link, View an Archive Site. This opens a pop-up window, and you can see all the different archives that are still available on the browser. So all of these archives still have a browser page available. If you are working with an archive that is not in this list, don't worry, the data is still there. It's just available via our FTP site. So there won't be a browser interface, but the data is still there. To close any pop-up windows, you can just click this check icon in the top right-hand corner, or you can click anywhere outside the window. Okay, so if we focus on the left-hand corner in the bottom now, again, we can see which release we're working with. So Ensembl Release 1.11, published in January 2024. Oh, and if you want to access any of our sister sites, at the very bottom, we have some links here. So to bacteria, fungi, plants, protists, and invertebrate metazoa. Or you can just open a new tab, which I'm going to do now, and simply type in ensemblgenomes.org. So this is the Ensembl Genome homepage. We've got the different portals. So we've got the coronavirus portal, rapid release. On the right-hand side, plants, invertebrate metazoa, protists, fungi, and bacteria. And if you open one of these portals, so I'm just going to open Ensembl Protists to show you how this looks like. You can see the main difference between the sites is this color scheme. So instead of the blue that we have in the main site, the Protists one is purple, but the structure, the format is still the same. So if you are working with any invertebrate genomes, the Ensembl Genome site would be your first point to go. Let me just put these two links in the chat so that you have them handy. Okay. So if we go back to the Ensembl main site, I've just scrolled back to the top of the page. Under the species-specific search, so under this blue box here, you can find out all the different genomes that we have available in this site. So on the right, you can see we have the section favorite genomes. If you have an account with us, you can actually change this. So just click on this pen icon, and you can change these genomes to your favorite genomes. On the left-hand side, you can also view a full list of all species. So I'm just going to click that link. And this sends you to our species page. And the species page has a list of all the species we have available in Ensembl, in the Ensembl main site. So for each species, we give you the common name, we've got the scientific name, taxon ID, assembly ID or assembly name, accession ID. And here on the right, you can see what data we have available for the species. So for example, for the giant tortoise, we've got a full gene build available. So this means we have full annotation data available, but we don't have variation or regulation data. If there is variation data or regulation data available, you would find a Y for yes in this column. You can search the column by scientific name or by common name. So let's say you want to look for Homo sapiens, simply type in the name, and that will filter your table down. So for human, we can see we've got the full gene build, we've got variation data, and we've got regulation data available. If you want to find out more information about the species, you can click on the common name. This brings you to the human species page, and here you get even more detailed information about the different data we have available for human. So we've got information about the genome assembly here on the left-hand side. We've got gene annotation data. For each of the types of data, you can also see an example. So for example, if you want to see how a gene for human looks like, you can click on example gene. We've got comparative genomics data, variation data, and regulation data. So again, for each of the types, you can see or view examples of the different features. If you're working with an older assembly, so let's say you're working with the GRCH37 assembly of human, you can access these through the species page as well. So under genome assembly here on the left, under other assemblies, there is a drop-down menu where you can select which assembly you want to work with, so GRCH37 or NCBI 36. Let's just select 37 and see how this looks like. And now you can see that this is the GRCH37 portal. Again, it's structured the same way as the main site, so you'll have no problem navigating it. Okay, so let's just look at some more information about the genome assembly for human. So let's click on more information and statistics. So this is the annotation information page for human. We've got a description of the assembly. We've got any patches here on the right, on the left-hand side. Again, links to the other assemblies we've got available for human, and a description of the gene annotation. On the right-hand side, we've got some summary statistics about the assembly. So we've got the original submitters of the assembly. So this is the assembly ID and the sequence ID, the number of base pairs, when the annotation was started, when the last update was done on the annotation. We've got the database version. Underneath, we've got some summary about the gene counts as well. So for example, the number of coding genes, non-coding genes, and so on. So if you want to find any information about the assembly, the genome assembly, you can find all of this in this page. If you want to go back to the homepage, you can just click the Ensembl icon in the top left-hand corner, and that just brings you back to the homepage. Okay, so this is all I've wanted to show you for this demonstration. Are there any questions so far? Okay, so I can see there's actually one question in the living document, and the question is, what is the difference between variation database and regulation database? So this is a really good question, especially because we're not going to go through this today because we won't have time. But So for each species, or for a number of different species, let's use human as an example. We have the gene annotation data, but we also have a separate database of any genetic variation in the genome. And then there's another database of any regulatory elements. So any features in the genome that control gene expression. So for example, if we go back to the full list of species and just click on the variation database column, this will sort the table and show you all of the species that have variation data available. So for example, we can see we have variation data available for American mink, Atlantic salmon, and so on. But actually, for American mink, we don't have any regulation data available. But instead, for Atlantic salmon, we do. So the Y just means yes, and the dash means no. I hope that makes sense. I will also enter the answer into the living document just so you have a reference for it. Okay, if there are no questions so far, for now, let's move on to the exercises. So if you go back to the training page and open the tab that says Species and Genome Assembly, let me just show you how this looks like again. So I'm back on my browser, back on the training page. If you scroll down to the bottom of the page under Species and Genome Assemblies, we're now going to do the exercises for this module. So there are a few exercises there. Please do them because they are really useful to become familiar with the site structure and where to find information about each of the genomes. You don't even have to do this specific species. You can enter your species of interest. So for example, if you were working with, I don't know, dolphin, you can switch panda to dolphin and search information about dolphin. Okay, so I'll give you, let's say 15 minutes for this, and then we'll come back together and move on to the next module. If you have any questions in the meantime, please feel free to enter them into the chat or the living document, and I and Ben will answer them for you. Okay, back to exploring genomic regions. So for this part, for this module, we're going to look at how we can visualize genomic regions on the browser. So I'm going to go straight to a demonstration. So I'll move to my browser in a minute to show you how you can view this genomic region in human, and then how to configure this page to see the different types of data that we're interested in. So just from earlier, in case you've probably missed it. So if we go to the training page, so Ensembl workshop, Ensembl browser workshop, Noblekinmat, you scroll down, exploring genomic regions, and you can copy the coordinates from the demonstration. And then you can open a new tab. I already have one open, so I'm back on the Ensembl homepage. And then you can select human from the species-specific search. So I'm going to use this because it's a bit more efficient to search Ensembl this way. And then we can enter our coordinates underneath in this text box. And Ensembl coordinates have a very specific format. So they start off with the chromosome number, followed by colon, followed by the start position, hyphen, and position. If you have a comma in your positions, you don't have to remove them. Ensembl will just ignore them. So if you have commas in the positions, just leave them. You can still search for coordinates that way. Okay, so let's just click on go. I'm just waiting for Ensembl to load. I'm so sorry to hear this, everyone, for those who are being abused in the chat. I'm very disappointed as well. Okay, so we can see that Ensembl is still loading. Let me just check if everything's okay. Every time I'm about to check something, it loads. So that's always very good. All right. So we've just searched for coordinates in human. And what we're seeing on the page now, which is still loading, this is what we call the location tab. So on the left-hand side, you can see that we have this tab open, location. And as we look at more data in Ensembl, as we view different genomic features, you'll see these tabs being populated. So in the afternoon, we're also going to look at genes and transcripts. So we're going to open a new tab called the Genes tab, and then another tab called the Transcript tab, and so on. So if you're ever not sure which page you're on, you can always look at these tabs. On the left-hand side, we've got what we call location-based displays. So these displays are all based on the location. So by default, we land on the region in detail view. So that's what you're seeing here on the right. So on the right, we have the region in detail view in 3 different resolutions. We've got the chromosome view up here, which is still loading. So I'll just leave that loading in the background. We have the region overview. So this is an overview of the region, which is highlighted in this red box. And it's 1 megabase wide or big. So you can see that here. And underneath, if you scroll to the bottom, this is what we call our region in detail view. So this is the region. This is only the region that we've searched for. Let's go back up just to go through each of the regions, each of the resolutions. So in the chromosome overview, we can see our region that we've searched for is highlighted in this red box here. Again, in the region overview, it's highlighted here, but here we have more information available to us. So in this view, you can find different tracks, and tracks is a genome browser specific term. And basically what it means is it's features mapped to a genomic region. So we've got these different features. We've got contigs, genes, regulatory build, and at the very bottom, we have a gene legend. So the different colors, what they mean, you can find underneath in the legend here. So let's just focus on this genes track. In this genes track, you'll notice that you have these rectangular shapes, and these rectangular shapes are the genes that are found in this region. So for example, I'm just highlighting this purple one here. This is the IL-21 IL-21AS1 gene. This golden one here is IL-21. Another golden one here, BBS12, and so on. And in the afternoon, we are going to look at different types of genes and transcripts and how they differ. So don't worry about the different colors just yet. I will go through them in the afternoon. Okay, so let's scroll down. Down here, we have our region in detail view. So here you're seeing our search region, but in even more detail. So instead of just seeing genes, you are also seeing the individual transcripts. And again, we're going to go through this in the afternoon to see what the different types of transcripts are. But the really nice thing about this view is this is completely interactive. So you can scroll along the view, or you can highlight a region. I am zoomed in quite a lot in my browser, so some of the settings or options you can't really see them. So let me just show you how this would look like on your browser. I'm just waiting for this to load. Whenever you see this Ensembl icon, that just means it's loading. So when it's like kind of grayed out like this, you just need to be a bit patient and wait for it to load. In the meantime, I'm just going to open a new tab and let it load in the background. Oh, okay. So it's loaded now. The view isn't there yet, but what I wanted to show you here mainly is are these options. So here on the right-hand side, there's an option that changes your mouse action to drag or select. So if you want to highlight a region or select a region, you'd select this one. And if you want to scroll along the genome, you can select this. And instead of highlighting a region, um, it will move the region left and right. If you notice that it's taking time for this to load at all, just refresh the page. That should reload it again. Yeah, especially when you see this icon here. So that means the image is broken. So again, just reload that page there. Right, we're almost there. Okay, so we've got our chromosome overview back up here. Our region overview is still loading, and then our region in detail view has just loaded. All right, so on our region in detail view, again, we have these different tracks showing you different features that are mapped to the region. But one main thing I want to show you here is these tracks are actually divided up into two separate, not regions, two separate ways. So all of the tracks that are located above the context track are features located on the forward strand, and all the features located underneath the context track are features located on the reverse track. Again, don't worry about these colors too much just yet. We are going to look at them later on in the afternoon, and also what the boxes and lines mean. So you can change the order of these tracks. So if you hover with your mouse next to the track name, you get this arrow, and you can actually move it around, move the track to put it closer to other tracks that you might want to compare. If you're not sure what information the track is showing you, you can always click on the track name. Oh, sorry. Not loading quite right. It hasn't loaded quite right. There we go. So I know it hasn't loaded quite right because the icons aren't here yet. But what I mainly want to show you here is you can click on the track name, and it will give you the right one. Oh, sorry. The view hasn't loaded quite right for me. So if you click on the track name, I've just clicked on genes there. You can find a description under this eye icon. So this track gives you the GenCode comprehensive set, which is gene set for human and mouse. There are also different settings on this pop-up window, so you can change the style of your track. You can generate a URL and share this with your collaborators if you want to show them something interesting. You can delete a track, or you can highlight the track. There are also ways to add more tracks to the view. So let's say you're actually interested in annotation from other databases, or you want to visualize any genetic variants in this view. You can add tracks by clicking Add or Remove Tracks just in the top left-hand corner. Or you can also click on Configure this page in the far left. So these two buttons do the exact same thing. And this will open a pop-up window. And in this pop-up window, you will find all the different tracks, all the different features that you can map to the genome that are available in Ensembl. So I'm just waiting for this to load. Oh, sorry about that. So it's loaded now. So this is the pop-up window. By default, you land on the active tracks. So that's what you're seeing here on the right-hand side. These are the tracks that are already turned on. You can turn these off by clicking on the square next to the name, or you can also see a description of each of the tracks. So if you click on the eye icon in the far right, you can find more information about the track. There are different categories that you can find here in the left-hand panel. So we have tracks relating to the sequence or assembly, genes and transcripts, mRNA and protein alignment, variation, regulation data, and also comparative genomics data. You can also search for a track. So let's say you want to visualize UniProt annotations on the genome. So you can type in UniProt. So UniProt is just another database that gives you information about different proteins. You can type in UniProt in the text box, and that will give you here on the right all the tracks that have the word UniProt in the name. So let's just turn on proteins from UniProt KB. So let's click on the square, select the style that you want. I'm just going to select normal. And you can see in the background, it's just reloaded the page there. If you want to turn off a track, again, just click on the name, on the square, click on off, and that will remove it. If you have an account with us, you can also save your tracks. So if you're using tracks quite often, that's very useful. You can find them then in the favorite track section here on the right. Okay, so let's close this menu and see how many proteins from UniProt we can find in our region. So again, this will just reload our view. Okay, so that's been quite quick. So we've got here, under here, we've got our new track that we've just added, proteins from UniProt KB. And sometimes you would have a second one as well. So if you scroll down, we have the second one here. So again, if you have two tracks with the same name, the track that's above contigs is the track that shows you any features on the forward strand, and the track that's below the contigs track are any features on the reverse strand. So we can see we have 1, 2, 3 proteins in this view. And these features are also interactive, so you can click on the feature. Let's see what this protein is. Just waiting for the information panel to load. Okay, so this track is from UniProt. This feature is from UniProt. This is the ID that was given by UniProt. And you can also find links to the actual entry. So if you click the ID, that will open a new tab and show you the entry in the UniProt database. The really cool thing about this this view as well is that you can download the image. So if you want to include it into your report, you can export the image. So at the top, there is an option to export the image. You can select the format that you want. PDF is usually the best, the highest resolution, and then just simply download it to your local machine. You can also share your view, your region view. So if you want to share any interesting tracks with your collaborators, for example, you can generate a URL using the share button here. And that will give you a URL here, which you can then copy and send to your collaborators. So some of you are might wonder, okay, why can't I just copy the URL from the browser address box? If you do that, your collaborators might not be able to see the exact same configurations that you've done. So for example, in my view, I've added the UniProt track here. And if you copy the URL from your browser address bar, they will not be able to see the UniProt configuration. So instead, we always ask you to generate a URL using this button here. And then finally, if you want to export data, so instead of downloading the image, if you want to export the actual data that you can see and hear, you can do that as well. So for example, all of the annotations that you're seeing in the view, simply click on Export Data here in the left-hand side. Just waiting for the pop-up window to load here. So in this pop-up window, you can again select, just like with the image, select what format you want your data to come in, and then you can download it to your local machine. But I just want to show you how this looks like. In the meantime, there was a question in the chat. So the question is, how do we know that the upper track is for forward and the lower track is for the reverse strand? So this is just the way that Ensembl, the visual view of the genome is built in Ensembl. But there are also different ways to find out whether your feature is located on the forward and reverse strand. And it's by looking at the feature name or the actual symbols in the feature. And I will show you the different ways during the presentation in the afternoon. So during the genes and transcripts module. Okay, so it's loaded now for me, the pop-up window. So I mentioned you can select the different formats that you want to export your data in. So for example, you can download the FASTA sequence only. You can download the annotation data. You can download it in different formats, so BED format, GFF, GTF, and so on. And here you can also change your coordinates. So if you realize actually you want to view a bigger location, you can change that here. And you can also highlight if you want to download any upstream or downstream sequences from here. Okay, so this is all I've wanted to show you for this view. We do have time for exercises now. So if we go back to our presentation, and we're going to move on to exploring genomic regions. So on the training page, please open the exercises the exercises tab and try those out for you. It might take some time to load, so I'll give you time until 12 o'clock for this. In the meantime, I will paste the questions into the chat as well in case the training page doesn't load for you again. So please try the exercises. We'll come back at 12 and just, and then move on to a lunch break. I believe. Okay, so it's almost 12 o'clock now. Um, so I know there wasn't much time to do the exercises for the Exploring Genomic Regions module. But please don't forget that the training page and the living document is available to you, will be available to you indefinitely. So you can always go back and try the exercises out in your own time. Just before we finish up for the morning, just want to bring your attention back to the agenda. So we're going to come back together using the same link at 2:30, and we're going to talk about genes and transcripts. And I'm going to introduce you to one of our most popular tools, which is BioMart. I will use the same link for today, but I will need to turn off all of the permissions. So if you want to ask any questions in the afternoon, please ask them in the chat or enter them into the living document. But for now, thank you for those who stayed behind. Again, I'm very sorry about the disturbance during the workshop, but hopefully in the afternoon we'll have everything sorted and we can relax and actually learn about Ensembl. So please be back in the same link at 2:30, and I wish you a wonderful break. Okay, welcome back, everyone. I hope you had a restful break. So we are going to continue on with our Ensembl workshop. This afternoon, we're going to look at how genes and transcripts are annotated in Ensembl, and I'll show you how you can view information about genes and transcripts on the browser. And finally, for our final thing in the workshop is we're going to look at a tool called BioMart, and this allows you to export large amounts of Ensembl data without the need to know any programming language. And at the end, we're going to just wrap everything up, summarize everything we've learned today, and I will give you some time to fill out a feedback survey. And so unfortunately, I had to remove all of the permissions for participants today. So for the afternoon, if you do have if you do have any questions, please put them in the chat box on Zoom, or you can enter your question in the living document on the training page. So just to recap from this morning, the training page, I will paste the link into the chat. So I am on my browser on my screen right now. So this is the training page we are using today. On the right-hand side, under materials, we have a link that says living document. And this is just a Google Doc, which you can refer back to in the future. But on page 4, we've got a section for questions. So if you have any questions or if you have any longer questions, you can enter them in here. And this document will be available to you indefinitely. So if you've forgotten what the answer was to your question, you can always come back to this. I suggest you keep the training page open on your browser because we're going to come back to it for the exercises after our module. So let's begin with our first module of the afternoon, which is genes and transcripts. Okay, so let's talk about how we actually annotate genes in Ensembl. Genomes that are annotated with genes are referred to as gene builds. And this is a term that you will come across quite often when working with genome browsers. There are 2 independent annotation methods that we use in Ensembl, but both rely on valid biological experimental data. So that means that we only add gene annotation when there is experimental evidence that a gene is transcribed. We have the Ensembl automatic annotation pipeline, which is applied to all vertebrate species in the Ensembl main site. And we also have the Havana manual annotation pipeline, which is done by a group of annotators, so people. And this pipeline is only applied to human, mouse, and partially zebrafish and rat. The automatic annotation method is a gene prediction pipeline. So we take known data, for example, transcripts, proteins, cDNA or RNA-seq data, and we map these to the genome. And it can take up to 2 weeks to annotate a single genome using the automatic annotation pipeline. The manual annotation method is done by an annotator, a person, so it can take several years in comparison to the automatic annotation for a single genome to be annotated this way. And this is just a list of the different annotation sources that we use in Ensembl, in our Ensembl annotation pipeline. For the automatic annotation, let me just change my mouse, sorry. For the automatic annotation, we retrieve nucleotide data such as cDNA, RNA-seq data that is submitted to the International Nucleotide Sequencing Database Collaboration, or INSDC. So this is a collaboration of the European Nucleotide Archive, ENA, NCBI's GenBank, and also the DNA Data Bank of Japan, DDBJ. We also retrieve proteomics data from SwissProt and Ensembl, and if there is not enough experimental data for a lesser studied organism, for example, we also perform annotation based on gene prediction by sequence similarity of a closely related species, closely related and better studied species. In the manual gene annotation pipeline, the annotation is done by a person on a case-by-case basis. So what they do is they look at specific data sets. So for example, RNA-seq data, transcript structure data that has been published. And the annotator also has a much wider range of evidence that they can retrieve from because they're doing the work manually rather than the automatic annotation, which is a pipeline. So it needs very specific types of file formats to be read. So that means for the manual annotation, there is more flexibility compared to the pipeline, compared to the automatic pipeline. The manual annotation process is much more laborious as the annotator moves along the genome to map any published experimental data using different databases. And because of this, it can take half a day to annotate a single gene. However, manual annotation is more comprehensive and also more accurate for difficult feature annotations. So for example, splice sites. But it also takes much more time. And this is why we only perform manual annotation on human, mouse, and partially zebrafish and rat. In Ensembl Genomes, so the browser that is dedicated to non-vertebrate genomes, the annotation process is done a bit differently. We don't do the gene annotation ourselves, but rather we rely on communities who are working with a particular species to do the annotation and submit this information to public archives. The annotation process by these groups can be quite similar to Ensembl's annotation process, but so they may use automatic annotation methods, manual annotation, manual curation methods. They may annotate by homology or by looking at open reading frames in the sequence. These are just a few species that you can find in the Ensembl Genomes browser and the different institutes and groups that produce the annotation data. So for example, for wheat, Triticum aestivum here in the top left, the annotation was produced by the International Wheat Genome Sequencing Collaboration, IWGSC, and they used automatic and manual curation methods. So this differs by the species. And if you want to find out which institute performed the annotation for the species in Ensembl Genomes, all you need to do is go to the species page and you'll see which institute or which group provided the annotation. Okay, so let's look back at the region in detail view, which we explored a bit during our second module this morning. So on my slide, what you're seeing is a snippet of that region in detail view. But what I want to show you here is how you would see a gene on the Ensembl browser. So on my slide, we've got the MAGT1 gene, and you can see down here that there are actually 5 different annotations for MAGT1. So we've got number 1, number 2, 3, 4, and 5. And the reason why you're seeing multiple annotations for MAGT1 is because in Ensembl, a gene is defined as a collection of transcripts. So we know that many alternative transcripts can be transcribed from a single locus, and this is what you're seeing here. So you're seeing the individual transcripts of MAGT1. And each transcript can be expressed differently and can also serve a slightly different function. So we have different types of transcripts, and we also color code them in Ensembl. So we've got gray, or I'm not quite sure what this color is. Yeah, I'd say this is gray. We've got a red transcript, blue, and this golden transcript here. So let's just go through each of them and what the difference is between them. So for the first example, we have we show you non-coding transcripts. So these are transcripts that don't produce any proteins, for example, RNA genes, and they're always color-coded in gray. We also give you information whether a transcript is protein coding. So that's the red one here. And these are transcripts that are translated and that do produce proteins. We also have non-coding transcripts of coding genes. So these are transcripts of coding genes, but they are not translated. For example, retained intron transcripts. They can be fragmented, or they might be missing a translation start site, or be faulty, for example. These are all colored in blue. And then finally, we have what we call the golden or merged transcript. And these transcripts represent the confidence in the annotation. So transcripts that are colored in golden mean when a transcript is colored in this golden color, it means that we have a high confidence in the annotation. But I'll talk about it more in the next slide. So these are the different types of transcripts. So now let's look at the individual features of a transcript. So if we just focus on this very first transcript of MAGT1, so we can see the color here. This is a golden transcript. So that means our confidence in the annotation is very high for this. But if you look closely, you'll notice that the transcript is made up of these boxes and these horizontal lines. So these boxes and lines just simply represent which regions are exonic and intronic regions. So anything that's a box is an exon, or in this case, this is quite a small exon, so it's just represented as a line here, as a vertical line. And anything that is a horizontal line is an intronic region. Next, you'll notice that some of the boxes are actually empty and the other boxes are shaded like this one. And that just tells you whether the exon is non-coding or coding. So this first example here that I'm highlighting with my mouse, this is a non-coding exon. And in the second transcript towards the end, we have a coding exon. So this is filled. Then you will also see, if you look really closely, you will also see that these horizontal lines actually have 2 different shapes. So this very first shape is actually it dips down. So it's like a valley or a V shape. But if you look at the COX7B gene up here in the top right-hand corner, you'll notice that these horizontal lines are actually more like hill-shaped. And what they mean is they just tell you in which direction on which strand the gene is located on. So whether it's on a forward or on the reverse strand. So if the gene is, or if the transcript is located on a forward strand, you'd have this A shape, this hill shape. And if it's located on the reverse strand, you'd have this V or valley shape. Another way to find out if the feature is on the forward or reverse strand is by looking at the name itself. So if you look at MAGT1, again, this golden transcript, next to the name, we have this less than sign, and that means that it's located on the reverse strand. And for COX7B, we have this greater than sign. So again, that means that it's located on the forward strand. So we have different levels of confidence in the annotation of genes and transcripts, and we always give you, or we always flag the annotation confidence in Ensembl. So I mentioned golden transcripts. These are transcripts that have been annotated identically by both the Ensembl annotation by the Ensembl automatic annotation pipeline and the Havana manual annotation pipeline. So this means that features features of the transcript that have been annotated identically by two independent annotation methods, so the automatic and the manual annotation. If they have been annotated identically by these two independent annotation methods, it means that there is a higher confidence in the function and also in the quality of transcripts, because you have two different pipelines predicting the same function for a single gene. So it just means that we're more confident in the annotation. We also have what we call the GenCode gene set. So the GenCode project focuses on the Ensembl human and mouse gene sets, and it is the merged gene set of the Ensembl automatically annotated and the Havana manually annotated genes in human and in mouse. It's also the default gene set used by many major projects, such as NOMAD. We've got the 100,000 Genomes project from Genomic England, and also the 1,000 Genomes project. Another project to know, or another flag to know about, which also dictates the annotation confidence, is the main select project. So main stands for matched annotation from the NCBI and Ensembl EBI. And it is just another measure of the quality of a transcript. So NCBI have their own gene annotation pipelines, RefSeq, and main select transcripts, so transcripts that have this main flag on them, are 100% identical in both RefSeq and Ensembl. So the main select transcript is also agreed to be the most biologically relevant and representative for the gene. So again, you have two independent annotation methods predicting the same annotation for a single transcript, meaning that there is more confidence in the annotation. And then elaborating on transcripts even more, I also want to introduce you to canonical transcripts. So these are a single representative transcript identified at every locus in every species. These transcripts are the most highly conserved, most highly expressed. They're in concordant with other transcript scoring algorithms and also uniprot canonical isoforms. They're also the longest coding sequence and have the largest number of clinically important variants. And wherever available, the Ensembl canonical transcript is usually also the main select transcript. Okay, so all features in Ensembl have an Ensembl stable ID assigned to them. So whenever you see an ID that starts with ENS, you can be sure that this annotation came from Ensembl. The ID is composed of the ENS prefix, followed by a single letter, which represents the feature type. So for example, G for gene, T for transcript, E for exon, and so on, followed by an 11-digit unique number. Stable IDs don't change between releases. If there is a minor change, we increase the version number after the dot. For any non-human species, we also add a 3-letter code between the ENS prefix and the feature type. So for example, for mouse, mus musculus, the ID would start with ENS, MUS, and then the feature type. And finally, I want to talk about gene ontology or Go terms. So just like in any language, there can be multiple terms for the same biological function of a gene. For example, one person might call it innate immunity, while another person would call it non-specific immunity. We humans know that we're talking about the same thing, but machines aren't quite that smart yet. So to tackle this, the scientific community came up with Go terms. And these are just a standardized way of describing a gene function to make sure that we are indeed talking about the same function, and also to make it more machine readable. Go terms are made up of a unique Go ID. So these usually start with Go followed by a colon. And it also has a human readable description. And you can see that Go terms are structured in a hierarchy. So a Go term can have a parent term, which describes the wider function of the feature, but it can also have child terms, which describe more detailed functions. There are 3 categories or aspects, as we call them, of Go terms. So the biological function describes what the protein does. The molecular function describes how the protein does it. And the cellular component describes where in the cell the protein is located. So if we take human cytochrome C as an example, the biological process is oxidative phosphorylation. So what does it do? How does it do it? It does it by oxidoreductase activity. And where is it located? It's located in the mitochondrial intermembrane space. So whenever we have this information available, we give it to you in Ensembl. And apart from Go terms, we also annotate protein domains. If it's a protein coding gene, we highlight any genetic variants if we have variant information for the species, and also any associated phenotypes. And we always provide links to external databases where there is an entry available. Okay, so I'm going to move on to our first demonstration for the afternoon. We are going to look at the human UQCRQ gene, and we're going to find out information about the gene and also its transcripts. Before I move on, I'm just going to check if there are any questions in the living document. No questions so far, and I don't see any questions in the chat. If you do have any questions, please feel free to enter them in either. Okay, right. So I'm back on my browser, and I already have the Ensembl browser open. But if you don't, you can just enter ensembl.org in your browser address bar, and you should be able to see the homepage like this one here on my screen. So to search, I'm going to use the species-specific search again in this blue box here. So first we need to select our species, in this case, human, and our gene of interest is UQCRQ. Oops, UQCRQ. And then you can hit go. You can also see we give you some direct links to these, but I just want to show you how the results will look like. Okay, so this is the results page for UQCRQ. So in our previous module, we searched for coordinates. And because we searched for specific coordinates in human, we were actually sent to the location tab straight away. But in this case, in our results, because we searched for a term, in our results, we're getting all of the entries that mention UQCRQ. So we're not just getting the results for the human gene, but we're also getting results for the human transcript. So that's what you're seeing here. Oh, and also any phenotypes that mention UQCRQ. But we're interested in the gene, so I'm going to select this very first hit in my results. And you can see, if you look in the top of the page, we have this new tab open, which we call the Gene tab. So we've got the Gene tab open here, and earlier today, we were looking at the Location tab. If you want to find any gene-specific information, so for example, the sequence of the gene, the function, and so on, you want to do this in the Gene tab. But if you want to find information about the location, the region, so for example, if you want to visualize the region, you'll need to switch to the Location tab for that. But don't worry about it, you'll have time to explore this during the exercise. Okay, so we are on the Gene tab. By default, we always land on the summary page, on the summary display here on the left-hand side. And that's what you're seeing here on the right. So we've got the gene name, we've got the Ensembl gene ID, we've got the description. So our UQCRQ gene is a ubiquinol cytochrome C reductase. We've got any synonyms. So remember, genes can have multiple names, so we always provide you with synonyms whenever available. We've got the location, which strand it's located on, about this gene. So this gives you, if you click on Show Transcript Table, this gives you the individual transcripts of this gene. So the UQCRQ has 6 different transcripts. But we're going to close this table for now because we're just going to focus on gene information first. Underneath, we have some links to external databases. So for example, a link to the HGNC symbol. We've got a link to UniProt and RefSeq as well. But most importantly, what I want to show you here is under gene type, we can see what type of gene this is. So this is protein coding. And under annotation method, how this gene was annotated by us. So we can see this annotation includes both automatic and manual curation methods. Underneath, we have a snippet of the region in detail view. So here you can just see the gene in context to the genomic region. UQCRQ is highlighted in this green color. We can see there are a few neighboring genes near UQCRQ as well. And if you might have noticed that the UQCRQ entries are above this context track. So that means it's located on the forward strand, which coincides with the location up here. Okay, so let's just explore a few different gene-based displays here in the top left-hand corner. Let's look at the sequence of the gene. So I've just clicked on sequence. And on the right-hand side, if you scroll down, this is a FASTA of our UQCRQ sequence. And we know it's in the FASTA format because it starts off with a greater than sign, followed by a description of the sequence, followed by the sequence itself. You'll see a legend just above the FASTA. So any UQCRQ exons are highlighted in this red color, and all exons in the region are highlighted in this peach color here. So what you're seeing in the FASTA file is you're seeing the UQCRQ gene, but you're also seeing neighboring genes. So this is what I'm highlighting here with my mouse. Anything that is not in red are neighboring genes. So we're also seeing some flanking regions of our UQCRQ. You can configure this page and make it a bit more helpful to you. So if you click on configure this page on the left, we can actually select how much flanking sequences we want to see. So if you just want to actually view the UQCRQ sequence, we can just change that to 0. You can also, for example, show variants or visualize variants within the sequence. So let's just turn this on and see how this looks. And what I also like to turn on are line numbering. So it makes it a bit easier for you to find where in the sequence you are at the moment. So you can select line numbering relative to the sequence or relative to the coordinates, so the entire genome. If you want to save your settings, just click on this check icon in the top right-hand corner or anywhere outside the window. So now we're just waiting for our legend to load here. Okay. While that's loading, I can show you a few other options you can do in this view. So you can actually already see that the line numbering is already turned on. So we can see that here on the side. You can download the sequence to your computer if you like as well. So you can click on download sequence up here or on download sequence here on the left-hand side. Select what format you want. Do you want it in FASTA format or do you want it in rich text format, RTF? So RTF is just downloading the file with all the different colorings that you're going to see in a minute. If you're not sure how these different file formats look like, we give you some examples at the very bottom of the pop-up window. So there's just a preview of each of the formats. So this is the FASTA format, and this is how it would look like if you downloaded the RTF. I usually download the compressed file simply because it just makes it a bit faster to download. But you can also preview the sequence before you download it as well. So it's already loaded, but it had to reload the page. So I'm just waiting for the legend to come back here. All right. Oh, okay. So during the configuration, if you remember, I turned on to show all variants within the sequence, and that's what you're seeing here at the moment. So in the legend, the different colors give you the different types of variants. So we've got 3' and 5' UTR variants, missense variants, variants located within splice regions, and so on. And that's what you're seeing here down here. If you're interested in one of the variants, so for example, let's say this missense variant here, you can just click on the variant, and that gives you a pop-up window with more information about the variant here. So if you were to download the RTF file, the rich text format file, you would be seeing all these different colorings with the links in them. Okay. Oh, yes. And the nice thing about this view as well is you can perform a blast search from here directly. So if you wanted to perform a blast on a specific region of the sequence, it's not working at the moment. I think I just have a few too many configurations on. So let me just turn these off. So you can highlight a region. Oh, I think we might have changed this. So we used to be able to just highlight a sequence and it used to give us a pop-up for a blast. It's not working at the moment. I'm not sure why, but I will talk to my colleagues about this. In any case, if you do want to perform a blast search, you can also just copy the entire region, the entire file. Ah, there it is. Okay. You can highlight the sequence. It gives you this pop-up window and that allows you to perform a blast search straight away. So you won't have to copy paste it. You can just highlight it from here. All right. Okay, so let's look at some other views in this gene tab that we're in. So if we go back to the top left-hand corner in our gene-based displays, we also have the section called ontology. So here you can find information about the function of the gene. So for UQCRQ, we have cellular component and biological process information. So if you just click maybe biological process to see what functions we've got associated with UQCRQ. So on the right-hand side, you get this table of all the different Go terms or gene ontology terms that have been assigned to UQCRQ. So we've got the ID, we've got the term, so the description, what it is, the annotation source, we've got the transcript. So Go terms are always associated with a transcript rather than the gene. So that's why you're seeing individual transcripts here. And you can also find some shortcuts to view this or to search this Go term on BioMart, which we will explore a bit later, or view it on a karyotype. Oh, yes. And then one last thing I want to show you for the gene tab is external references. So in bioinformatics, you would often use multiple databases to look at information or to explore more details about a genetic feature. And under external references here, we give you a list of external databases and also a direct link to each of the entries for that. So for example, we've got a link to Expression Atlas, Reactome, NCBI, Wikigene, and so on. And you can explore this directly from Ensembl. So you don't have to go to the separate database. You can just click or search for your gene of interest in Ensembl and click through it from external references. Okay. So next, I want to show you what transcript-specific information you can find. So if we open the transcript table back up, we already explored this a bit, but we know that we have 6 different transcripts for UQCRQ. So 1, 2, 3, 4, 5, 6, and we have some more detailed information about them or about each of them in this table. So we've got the length. If it's protein coding, we also give you the length of the protein, the biotype. So this is what type of transcript it is, and some links to external databases. And if you scroll to the far right, we give you the different flags for them as well. So these are the flags that just describe how confident we are in the annotation. So for this very first transcript, this is actually a golden transcript. We can see that we have the main select flag. So this is the matched annotation between NCBI and Ensembl EBI. And we also have the Ensembl canonical transcript flag here. Let's explore more information about this very first transcript. So let's click on the transcript ID. Okay. So now if you look at the top of the page, we have this new tab open, which is the transcript tab. So here on the right, on the left, sorry, here what you're seeing is now transcript related information. If you want to go back to the gene tab, you can just switch on the gene tab. So just click on the gene tab and that will send you back to where you were before. So we are on the summary display right now. So this is similar to the gene tab, but what you instead of the region and detail view that we saw earlier, what you're now seeing is the transcript on its own. So you can see how the transcript is made up of these introns, so these lines, and exons. So remember, anything that is filled is a coding exon, and anything that's an empty box is a non-coding exon. Okay, so in this view, we can also view the sequence, but the transcript sequence this time instead of the gene sequence. So one of the views that I really find useful is the cDNA sequence view. So here on the right-hand side, we can see that our annotation from our variants is still turned on. So let me just turn that off so that it's a bit easier to see. So just click on configure this page, go to show variants, and then disable that. And now we have all the colorings gone, just to make it a bit easier. So what you're seeing in this view is you're seeing 3 different ways of the transcript sequence. So you're seeing the the cDNA, so the complementary DNA, complementary sequence. In the second line, you're seeing the coding sequence. And in the next line, you're seeing, or in the third line, you're seeing the amino acid sequence. So, which would always start with the methionine here. And this is really useful if you're looking, if you're, for example, looking at variants and you want to see which variant, where the variant is located in and which amino acid is affected by the variant. Another useful view as well is the exons view. So if we just click on sequence under exons, this sequence or this view is in a tabular form this time, and we're giving you the sequences of each exon. So we've got exon number 1, number 2, and so on. The sequences are also highlighted in different colors. The variant view is turned on again, so let me just turn this off. Okay. So I've just turned off the variants again. So in this view, you can see how each exon is made up. So the different colors you can find in the legend at the top. So anything that's colored in blue, in this blue font, it means that the sequence is translated. And anything that's colored in the orange font is any sequence that is not translated, so UTR, untranslated region. So this view is also very useful to see how whether an exon is partially translated or fully translated. Another thing I want to show you as well is the AlphaFold predicted model. So if you go under protein information in the left-hand panel and click on AlphaFold predicted model, what you're seeing here is the predicted protein structure for this transcript. And this view is interactive, so you can move the protein around with your mouse. You can zoom in, you can zoom out. On the left-hand side, you can highlight any specific regions in the protein. So it's a very nice way to visualize your protein, if you like. And then finally, if you go to general identifiers here on the left-hand side, again in the panel, you can, just like before, you can find links to external databases, to the entries of other databases. So if you want to view this transcript, for example, on ENA or PDB and so on, you can do so from this page. All right, so that's all I've wanted to show you for this demonstration. So now we have time for exercises. But before I let you go and do the exercises, are there any questions? Okay, no questions so far. All right, so if we go back to the training page, open up the genes and transcripts exercises. I hope that the page will now load for you. If it doesn't, please let me know and I can put the questions back in the chat. So I'll give you some time for this, maybe 15 minutes, and then we'll come back together to see if you need any more time. If not, we'll continue on with the next module. So please be back at 3:35. All right, it's 3:35 now. Are there any questions so far? All right, no questions in the chat, and I don't see any new questions in the living document either. I know it might not have been much time for the exercises, but I do want to move on to the next module because I really want to show you this tool because I think it will be very useful to you. Just don't forget that the exercises will still be there at the end of the day. They will be available to you indefinitely. So later on, if you have time, you can always come back and finish the exercises. But for now, let's just move on. So for this final module for today, I am going to talk about BioMart, which is one of our most popular tools, I'd say, in Ensembl. So BioMart is a web-based tool to export Ensembl data without needing to have any programming skills. You can build queries using a point and click method with your mouse, and you can generate custom data tables and files. But one thing that BioMart is it's not meant for downloading whole genomes. So if you want to download the entire sequence of a genome, for example, or the entire annotation set for a genome, you need to go to our FTP site for that. For Ensembl, you can use up to 500 entries at a time. If you do go over that, I do suggest that you separate it into smaller queries, just so that BioMart can actually process them efficiently. So why should you use BioMart if you can get all of the information that you need from the different Ensembl pages? So you can use the Ensembl website and go to the individual pages, just like how you could go to specialty shops to do your grocery shopping. But why should you make your life difficult if you can just go to a big supermarket and get everything that you need from a single site? So this is what BioMart is trying to be. It's trying to be this one-stop shop for all the different types of data you can get from Ensembl. In BioMart, you can query multiple genes or variants at once. You can convert any IDs. You can extract loci or coordinates and download multiple sequences in one go. So in other words, you can use BioMart for tasks that would be much too time consuming by going through the Ensembl browser alone. And you can find BioMart on Ensembl and so Ensembl main site, the site dedicated to vertebrate genomes, and also the Ensembl genome site, which is dedicated to non-vertebrate genomes. I mentioned earlier today that the structure of these browsers are very similar. So now that you're familiar with the Ensembl main site, if you are working with plants, you'll have no problem navigating the plant site. And you can find a link for BioMart in that navigation bar at the top of each website. So BioMart is available in Ensembl, in the Ensembl main site and Ensembl genomes. It's available for plants, invertebrate metazoa, protists, and fungi. But one thing you're going to see, or you might be missing here, is bacteria. So we don't have BioMart available for bacteria simply because there are too many of them to create a BioMart for it. All right, so now that I've introduced you to BioMart, let's look at how you can actually use the tool. So the easiest way to use BioMart is using this 4-step principle. So let's just look at each of the steps in a little more detail. The first step that you need to do is you need to define the database that you want to do your search in. So you can select from genes, variation, regulation, or mouse strain. And then you need to select what species you're working with, so your species of interest. In the second step, you are defining your set of genes or variants by applying filters. So these filters can be a region in the genome, a list of IDs, a particular function or phenotype. So this is going to be your input. And you can build simple queries by applying a single filter only. Or you can build more complex queries by applying multiple filters. So for example, let's say you have a particular region in the genome that you're interested in, but you only want to extract specific transcripts from that region. So you can apply 2 different filters that narrows down your search. And this leaves you with a more specific filter for which you apply attributes to. So attributes, this is our third step. What you're doing here is you're defining the type of data that you want for that set. So you're selecting the attributes of your data output. So for example, you can choose if your output should have things listed like IDs, features, variants, sequences, or homologs. And then finally, in the fourth step, once you're happy with the query, you can view and download your results in different formats. So the formats available to you are HTML, tab-separated format, comma-separated format, or Excel format. Just keep in mind, if you do open your table on Excel, just make sure that you turn off the automatic formatting, because sometimes Excel likes to automatically format gene names into dates. So just keep that in mind. And if you are downloading sequences, you can only get them in the FASTA format. Oh, and for those interested, we have 2 very interesting papers here. So the second paper just goes through each of the different BioMart steps in more detail. And the first paper just talks about how the BioMart was built. So if you're interested in the technical bits, these papers here are very interesting reads. Okay, so for this demonstration, we're going to look at a set of 6 human genes. So ESPN, MYH9, USH1C, CISD2, THRB, and BRCA2. And for each of these 6 genes, we're going to use BioMart to find out their NCBI IDs, their functions, so gene ontology terms, and also retrieve the cDNA sequences. So if you want to follow along, you can copy the list of genes from the training page. So let's move back to my browser. I am on the training page right now. And if you open the BioMart tab and click on demo, you can find the list of genes here. So I'm just going to copy those. Actually, I can see that one of the genes is different to our demonstration gene. So I'm just going to copy my list actually. All right. So next you want to open Ensembl. So I already have Ensembl open in my other tab. And we want to open the BioMart tool. On the top of the page, you can find a shortcut to BioMart. So I'm just going to click that link. Okay. So this is BioMart. This is the interface. And so just to recap, we want to follow 4 steps to make our BioMart query as efficiently as possible. First, we need to select the data set. Second, we need to select our filters. So this is our input. Next, we need to select our attributes. So what information do we want to get from Ensembl? And then we can look at the results. So let's select the database. We have a list of genes. So we're going to select the genes, Ensembl genes database. And our species of interest is human. So we're going to select human from this drop-down menu. And now on the left-hand side, we have more options available to us. So first step is dataset. Second step is filters. So if we click filters, here we can select or enter our input. There are loads of different categories that you can choose from. So for example, if you had a list of coordinates, you can enter them in region, or you can select specific chromosomes that you want to do your search in. If you want to select a chromosome, just click on the chromosome number, and that will add it to your list or to your filters here on the left-hand side. If you want to select multiple chromosomes, just press your command or control button on your keyboard and select all the different chromosomes you want to. And to remove the filter, just uncheck the box, and that will delete it from your filter here. There are loads of different categories, which I will let you explore during the exercises in your own time. So we have a list of 6 genes. So this is going to be under the gene category, which I will expand now. And you can enter your list of genes in the second option. So input, external references, ID list. I've already got my list of genes copied, so I just need to paste them into the text box. Your list of genes can be in the comma-separated format, which is what we're seeing here. It can also come, or you can also paste it in a tab-separated format or as a list, so 1 gene per line. Or you can also choose to upload a file if you have one on your computer. Next, you need to tell Ensembl or tell BioMart what type of IDs you've just given it, because it's not quite that smart yet. You need to tell us what type of IDs they are. So we've given gene names. So all you need to do is from the drop-down menu, select gene names and make sure that it's been added to our filters here on the left. Okay, so step 2 is done. We've given our input. Now we move on to step 3, where we select our attribute. So here we tell Ensembl what information we want from the database. So remember from the slide, we want to get the NCBI IDs, we want to get the Go terms, the function, and we want to get the cDNA sequences. Now, the attributes are divided up into different categories, fields. different types. So you can select one type at a time. So if you wanted to retrieve any features, so for example, IDs, you can find this under features. If you wanted to retrieve homologs, you can find this under homologs here. Any variant, genetic variant information under variant. Or if you want to retrieve sequence, you need to select this last bit here. So because we want to get features and we also want to get sequences, we actually need to separate our BioMart query into two parts, where we first find the features of our genes, and then we come back and get the sequences, because you can only select one at a time. So let's start with the features. Here on the left-hand side, we can see that we have a few features already selected. So if you open the gene tab here, you can see that the gene stable ID and the transcript stable ID is already turned on. You can turn these off if you want. So just click on the attribute that you want to remove, and that deletes it from your attributes list. The first thing I like to do is to make sure that my output will match my input. So you want to make sure that your output, that you know which output belongs to which gene. So in order to do that, we actually need to print the gene name in our output. So from the list, I can select gene name. So I've just done that here on the right-hand side. And that's added to my attributes list now. Okay, so next we want to select our NCBI ID, so we can find that under the external tab. So if you scroll down under external references, we want to get the NCBI ID. So all you need to do is search for it, description, NCBI ID. And the final thing we want is the go term, so the function. So that was up here. If you scroll back up, I'll get the accession. So this is the go ID. And I'll also get the name, so this is the more human readable part. Okay, and now here on the left-hand side, we want to check that we indeed have everything we want. So we've got the gene stable ID, transcript stable ID, I'll keep that, gene name, NCBI gene ID, and then the go terms. Now, one thing I usually do after step 2, actually, if we go back to filters, what I like to do in step 2, at the end of step 2, is to count the number of genes that my filter applies to. So this is just a little sanity check to make sure that you've applied the correct filters. So if I press this count button up here, I'm going to expect the number 6, because in our filters, we've entered 6 genes. So 1, 2, 3, 4, 5, 6. So let's just count. And indeed, you can see up here, our filter applies to 6 out of 70,711 genes in the human genome. So that's fine. So now we can get our results. So all you need to do is click on results in the top left-hand corner. And that was very quick. So here on the right-hand side, we now have our table, our output, including the different attributes that we selected. So the different information that we wanted from Ensembl. By default, we're only seeing the first 10 rows. So if you want to see the entire table, click on all. Under view, click on all, and that will open a new tab. Just might take a bit of time to load, and that will give you the entire table of your results, the entire output for your results. Okay, so we've got the stable ID, the transcript ID, the gene name. So this allows us to check which entry actually belongs to which gene from our input. We've got the NCBI ID, the go term, so the go term accession, and the go term name. Now, the first thing you're going to see is there are actually multiple results for each gene. So we've got in this first part, this D2. If If you scroll down, we have loads of entries for BRCA2, MYH9, and so on. So you're probably wondering, why do I have entries, so many entries for each gene? So if you look closely, you're actually going to see the results for each transcript. So you can, if we focus on the transcript column, we can see that there are actually multiple transcripts for each gene. But then you're also seeing you have multiple entries for each transcript. For example, for this first transcript, we have 2 entries. And this is because there can be multiple Go terms associated with a single transcript. So if we focus on this transcript ending 3-2, and we move down to the Go term accession column, we can actually see that there are 2 different Go terms associated with this transcript. And that's the same for all of the other entries here. So for the transcript ending 8-6, if you focus on the Go term accession column, you can see that all of these different entries are actually different Go terms. Okay, so let's close this table. If you want to download the results, so instead of viewing it on the browser, if you want to download the results, all you need to do is click on Export All Results to, select what type of file you want. So do you want it as it is or as a compressed file, or do you want it to be sent to your email? And then you need to select the format. So HTML, comma separated, tab separated, or Excel, and then just click go. Or if you wanted your results to be sent by email, you just need to enter your email address underneath here. Oh yes. And then if you want to share your BioMart results with your collaborators, you can also generate a URL really easily. So there is a URL button at the top of the page. This URL will be quite long, so make sure that you copy the entire URL and paste it. Otherwise, they won't be able to see. Otherwise, they'll be seeing an error message, I'd say. So make sure you copy the entire URL. Okay, so we've got our NCBI IDs, we've got our Go terms. So now let's get our cDNA sequences. So if you want to start a completely new BioMart query, that's fine. You can reset your query by clicking the New button in the top left-hand corner. Or if you're lazy like me, you can just go back to attributes and change your settings there. So let's just click back on attributes. And this time we want to select sequences. Okay, so let's start with the header information. So the description of the sequence. We've already got a few features selected. So again, here on the left-hand side, you can see we've got this gene stable ID and then the transcript stable ID. I'll just remove the version just for, so it's a bit easier to see. And the first thing you'll want to do again is to make sure that your output matches your input. So we're going to select the gene name. Okay, so our header is now selected. Now let's select the type of sequence that we want to get. So at the moment, we've got the peptide sequence selected here, but what we want is the cDNA sequence, the complementary DNA. So we're going to just select that from the list here. The nice thing about this view as well is in this image, it actually highlights what part of the transcript you're about to download. So if we, for example, select 5' UTR, you can see that only the 5' region is highlighted here. So it's just a little reminder which region of the transcript you're about to download. Okay, cDNA sequences, that's selected. Our output matches our input, so that's also good. So now we can click on our results again. Okay, so on the right-hand side, we now have our sequences in FASTA format. Again, we're only seeing the first 10 rows, so let's open the entire output. Okay, so here we've got now our FASTA file. It's quite big. You can keep scrolling down to the bottom of the page. You can see it's quite a big file. Again, for each uh, for each gene, you're going to get multiple entries. So if we just look at THRB here, so the third and fourth sequence in our file, um, these sequences, sorry, we give you the sequences for each of the transcripts. So that's why you're seeing multiple entries here. So this first sequence of THRB is for this particular transcript. Second sequence is for another transcript, and so on. So depending on how many transcripts each gene has, we'll give you the sequences for each of them. Okay. And if you want to download your file, similar to before, select how you want your file to be. Usually I just select compress because it makes it a bit faster, and just click go. Remember that for FASTA, for any sequences that you get from BioMart, you can only get them in the FASTA format. All right. So that was BioMart. It's quite straightforward as long as you use four-step principle. Makes it very efficient to find the data that you want from Ensembl. So now you have time to explore the tool on your own. So if you open the training page back up and open the BioMart tab, there are a few exercises in there. I'll give you 15 minutes for this. And then we'll come back together and just wrap everything up. And there'll be some time for some questions as well at the end if you have any. If you have any questions right now, please feel free to put them in the chat or in the living document. All right, so it's 4:15 now. So I hope that you were able to do some of the exercises for BioMart. I just want to go through some closing slides with you. And if we do have time towards the end, please feel free to stay on and ask questions or continue with the exercise. So for now, let's just go through some slides. So the first thing I'm going to ask you to do is to fill out the feedback survey for us. So we always ask participants at the end of the workshop to fill this out for us because it really helps us improve our courses and make it better for future participants. I'm going to put the link to the survey in the Zoom chat box, but you can also find it on the training page. So if you go to the training page, just above the exercises, there is a link for the surveys. You can open this. This shouldn't take any more than 3 minutes, so I'll give you some time for this now. But towards the end, if you want, you can choose to retrieve a certificate of attendance. So simply click yes, and then in the next page, you just need to enter your email address that you want it to be sent to. So I'll give you some time to fill this out now, and then we'll just summarize what we've learned today. Okay, thank you to those who have filled out the survey. I will keep it open for another week. So when I share the recordings, I'm also going to share a link to the survey. So for those who did not get a chance to fill it out, please do. But for those who have, thank you very much. Okay, so let's just summarize what we've learned today. So what are genome browsers? So today we learned that that genomic data are quite large and they're constantly changing. And genome browsers are there to visualize genomic regions in genomes. So genome browsers provide annotation. This includes genes, genetic variants, regulatory data, and so on. But what genome browsers are especially useful for is to get information about genomic features. So genome browsers allow you to search, browse, retrieve, and analyze genomic data. Ensembl, in particular, maps genomic regions to genome sequences that are publicly available. And what we do is we perform our own analyses to the data. Ensembl is also what we call an added value resource. So an added value resource just brings together information from a wide range of other databases into a single site. So it makes it a bit easier for you to find information about, for example, a gene. Now, remember that different genome browsers may use the same reference genome, but their annotation methods will be different. So the annotation might not be exactly the same between Ensembl and NCBI, for example. Today, we've only really looked at gene annotation data in Ensembl, but we also provide genetic variation data, comparative genomics data, so gene trees and homologs and so on, and also regulatory data for a number of different species. And how is all of this data organized? So we mainly looked at the main site today, the ensembl.org website, but you can also access non-vertebrate species through Ensembl genomes. You also have access to our DRCH37 portal, so the older human genome that is available, and you'll have access to our archives for up to 5 years. We also looked at BioMart, which is a tool that allows you to extract large amounts of Ensembl data using the browser. And for those who are more advanced, you can also access Ensembl data via our Perl API, REST API, and MySQL servers. And if you need to download any whole genome data, you can go to our FTP site for that. So there's a link on my slide here that's very handy for you to use. Now, if you did enjoy the course, please do recommend us to your colleagues. We do these courses around the world. So today we only really did a half-day course, but on our usual full-day course, we also go through comparative genomics, regulatory, and variation data. We also have more advanced courses where we show you how you can access Ensembl data via our REST API. And we also provide a train the trainer course. So this allows trainers from around the world to spread the word about Ensembl even more. If you are interested in hosting a workshop, you can go to our training page for that. So there's more information at training.ensembl.org/hosting, or you can email us directly at helpdesk@ensembl.org. And then finally, or no, not yet finally, almost finally, you can always reach out to us if you have any questions. So you can email us at the help desk email address, or you can get in touch with us on Facebook, LinkedIn, and X. For any news or job openings or just general updates in Ensembl, we always publish any updates on our blog at ensembl.info. Now, if you want to learn more about bioinformatics, so for example, if you're quite new to bioinformatics, there are some very useful resources on this slide here. So the EBI, European Bioinformatics Institute, has a training page. And this is really, really useful because there are a lot of different online courses there that you can complete in your own time. So for example, there is a course that's called Bioinformatics for the Terrified. So that one's very useful, especially if you're new to bioinformatics and just want to get an introduction to it. For any Ensembl related courses, you can always go to ensembl.org, or you can visit our YouTube for any tutorials for Ensembl data. If you have any specific questions at all and you can't find the answer online, please don't be shy to email us. So we are in charge of the help desk in Ensembl. There are real people behind it. So if you have any questions, please feel free to email us at helpdesk@ensembl.org. And now I mentioned Ensembl is completely open source. So the only thing we ask you to do is to cite us. So the paper you want to cite, if you're using Ensembl data, is always the most recent one. So the most recent paper is Ensembl 2024 here on my slide. And now, with that, I thank you everyone for being so patient, and I'm very sorry again about this morning. I hope it didn't disturb you too much. The recordings will be made available to you. I will try to finish them or finish processing them today. And we have a couple more minutes time. So if you have any questions at all, I'll stay on for a couple more minutes. And if not, please have a good day or evening, afternoon. And I hope you found the course useful, and hopefully see you soon. Thank you all very much.